All right, everybody. In this chapter, we're going to discuss the endocrine system. And so, what we've seen so far, we've noticed that cellular activity is able to be altered in the face of changing conditions, mostly in order to maintain homeostasis, right? Like when the concentration of calcium ions in the blood decreases, osteoclasts become more active, and they're going to resorb more bone to release more calcium ions. Well, how does a cell know how and when to alter its activities? The answer lies in the body's two major regulatory systems, and those are going to be the nervous system, which we've already covered, and now we're going to turn to the other one, the endocrine system. And these work together. Basically, these organs work by synthesizing and secreting chemical messengers called hormones into the blood. These hormones are then going to travel through the blood tissue to their target cells, and then these target cells are going to have specific receptors to which these hormones can bind, and then those are going to lead to very specific changes in cellular function. Most of these functions sort of revolve around maintaining fluid balance, electrolyte balance, promoting growth, regulating metabolic reactions, and responding to stressors. So in chapter 16, we're going to explore the organs and the cells of the endocrine system, as well as the hormones they produce, how these hormones maintain homeostasis, and then we're going to look at the impact of the endocrine system, as well as endocrine system disorders, and how often they very dramatically affect the body's functions. First things first, we need to look at an overview of the endocrine system, and this is going to be examining the differences in between nervous and endocrine systems, as well as the types of chemical signals that it uses, the anatomy of the endocrine organs, and then finally the main properties of those hormones, those chemical messengers that these endocrine organs produce. And so, as I said, these endocrine organs work together, secreting hormones into the blood. These hormones are going to be these long distance traveling messengers that travel to very specific target cells within target tissues, and they're going to influence their functions to basically maintain homeostasis. So the endocrine system and the nervous systems both complement each other, maintaining homeostasis of nearly every physiological variable. Both systems use chemicals to communicate with other cells. This is an example of the cell-cell communication principle. But the two systems differ significantly in how they regulate body functions. As we talked about for the nervous system, this is going to operate directly through a series of neurons, directly affecting their target cells via release of neurotransmitters. These effects of the nervous system are almost immediate, but they're relatively short-lasting unless the stimulation is repetitive. Now, the endocrine cells, these do not come in close contact with their target cells. Instead, I'll show you in just a second, these cells release those hormones into the interstitial fluid surrounding them, after which they will enter the blood. And when they do this, they diffuse through or between the cells of tiny blood capillaries, and then as the blood exits the capillaries, it transports the hormones, to the heart and through larger blood vessels called veins. And then after leaving the heart, the blood transports the hormones to the rest of the body through arteries. And eventually this is going to branch out repeatedly into smaller vessels and finally back into capillary beds. Once they're back in these capillary beds, far from where they started, the hormones will diffuse out of the blood into the interstitial fluid, and they'll reach the receptors on their target cells. Generally, these effects of hormones can require several seconds to several hours or even days to elicit their effects, but these effects are generally much longer lasting than the nervous system effects. Although working together, the nervous system and the endocrine system can work together to increase each other's effects. So here you see an image that shows the interstitial fluid down here surrounding a capillary. And so endocrine cells are going to release those hormones into the interstitial fluid. The interstitial fluid is then going to transport those hormones into the bloodstream, and they're going to diffuse out into the blood capillaries, and then all the way up through veins to the heart, get pumped out the heart, uh, into the lungs, out of the lungs, back into the heart, out of the heart, back into arteries. And as they travel through these arteries, they're going to make their way into eventually arterioles and then blood capillaries near their target cells. And so once they're near their target cells, these hormones are going to diffuse back out of the capillary bed into the interstitial fluid and then onto these receptors of their target cells. And so, again, this can take several seconds to several minutes, and they can last for days. The effects of these can last for days. Now, hormones are actually able to be classified into three separate classes. There are the classic hormones, the classic endocrine systems that are secreted into the blood and they affect long distance targets. And those are just regular hormones. However, there are also two types of local signals where the hormone is secreted into interstitial fluid and then it can affect local cells without ever actually entering the blood. And these could be paracrine signals and autocrine signals. And now paracrine signals are going to consist of a chemical secreted by cells into the extracellular fluid, and this is going to influence cells that are near it. But it does not ever enter those hormones do not ever enter the blood. In autocrine signals, the chemical secreted by the cells into the extracellular fluid elicits effects from the same cell. Now, although we primarily consider classic endocrine systems signals in this chapter, all three types are an important part of the body's regulatory system. And here you see a good diagram showing you the difference of these three pathways. And as I said, we're going to focus mainly on these endocrine normal pathways where the endocrine cell secretes hormones into the blood. The hormones travel to distant cells where they'll then elicit their effect. Then, but the other important ones are the paracrine signals, come out of the tissue into the extracellular fluid and then on some nearby cell that will elicit their effect. And then you also have autocrine, where the cell secretes a hormone into the extracellular fluid, and then it will latch onto receptors of itself, and it will elicit some effect of its own self. It's a sort of self-regulating hormone. I keep wanting to say neurotransmitter. Yeah, self-regulating hormone. Now test yourself. What type of signal is going to occur when a cell secretes a chemical into the extracellular fluid, and then this chemical affects nearby different cells? Is that autocrine, endocrine, or paracrine? That is a paracrine signal. Remember, that is a signal sent out from one cell into the extracellular fluid and affects different nearby cells. If it was endocrine, it would be distant different cells, and autocrine would be the same cell. Now, the main organs of the endocrine system are going to be called the endocrine glands. And I want you to notice that these are a very diverse group of organs located all throughout the body. Now, they share a common function of regulating other cell types through the production and secretion of hormones. And I want you to remember that endocrine glands are going to consist of ductless glandular epithelial cells that work to secrete their hormones into the interstitial fluid. in contrast to exocrine glands. And exocrine glands are going to secrete their products into ducts that lead to the body's surface or cavities. Think of the difference between a sweat gland and your pancreas. The sweat gland has a duct that leads outside. The pancreas is more of just that glandular, spongy material. And there are generally considered to be 7 primary organs of the endocrine system, as well as several secondary organs. Those 7 primary organs of the endocrine system include the anterior pituitary gland, and this is going to be situated in the sphenoid bone of the skull. And then there's the thyroid gland located in the anterior neck. Then you're going to have 3 to 5 small parathyroid glands located posterior to the thyroid gland. Then you have the paired adrenal cortices, and these are located on the superior surface of the kidneys. You have the endocrine pancreas located in the left side of the abdominal cavity, mostly posterior to the stomach. You have the thymus located in the superior mediastinum, and then that's usually located just above the heart in layman's terms. And then you have the paired ovaries or testes. Ovaries are going to be in the pelvic cavities of females, and testes are going to be suspended below the pelvic cavity for males. You also have secondary endocrine organs that are technically not considered part of the endocrine system. These organs do produce hormones, but essentially belong to other systems. I want you to note that the testes and ovaries are often considered secondary endocrine organs, as they are also part of the reproductive system. Many secondary endocrine organs are neuroendocrine organs, including the hypothalamus and the pineal gland. These are parts of the brain, and as well as the adrenal medulla in the core of the adrenal gland. Anatomically, these structures are all nervous tissue, yet they secrete chemicals that act as hormones. These are called neurohormones. Other organs and tissues that act like this are the heart, kidneys, small intestine, and adipose tissue. And then sometimes cancer cells can even act as secondary endocrine tissues. And we'll talk about that as paraneoplastic syndrome. So here you have an overview, essentially, of the entirety of the endocrine system, leading all the way from the hypothalamus, the pineal gland, the posterior pituitary gland in the brain, as well as the anterior pituitary gland. Down into the throat, you have the thyroid and the parathyroid glands. In the chest, you have the thymus gland, generally covering the heart. On top of the kidneys, you have the adrenal cortices and the adrenal medullas. You have the endocrine pancreas, located just behind the stomach. And then depending on your biological sex, you have either ovaries or testes. And all of these work to secrete hormones, long-distance chemical travelers in the blood. Now, as I said, the heart and kidneys aren't the only secondary endocrine tissues able to produce hormones. Cancer cells can too. The signs and symptoms of this paraneoplastic syndrome is a sort of collective accompaniment of hormone secretion from these cancer cells, with many different types of cancer cells causing paraneoplastic syndrome. But the most common are essentially lung and GI cancers, gastrointestinal cancers. And the effects really depend on the type of hormone secreted by the cancer cells. Commonly, you're going to find fluid imbalances, calcium imbalances, sodium imbalances, and the symptoms can often precede the symptoms of the cancer, and leading a general physician to investigate them earlier may lead to an earlier diagnosis. Now, these hormones, as I've said, are chemicals that are going to regulate some functions of other cells. A good way to think of them is they're kind of like middle management of the human body. They are going to take an order from their endocrine organ, and then they're going to go tell other cells what to do by stimulating some sort of change in those cells. Before we discuss all of the individual hormones, let's look at their general properties and several different types of hormones, how they travel through the blood, how they interact with their target cells, and how they work to produce certain effects. Let's also explore the mechanisms of hormone secretion and how it's regulated, and then how hormones may interact with one another and how they're eventually eliminated. Now, the classes of hormones include two basic chemical structure classifications. First, you have the amino acid based hormones that are going to be made up of one or more amino acids, ranging in size from one amino acid, and these are going to be your amine proteins. I'm sorry, your amine hormones. You have multiple amino acids making up peptide hormones, or even you'll have complete proteins making up protein hormones. Generally, these are going to be secreted by the anterior pituitary gland, the pancreas, the thymus, the thyroid, and the parathyroid glands. And essentially, these are going to be hydrophilic, freely interacting with water molecules and binding to the plasma membrane receptors of cells. Alternatively, you have steroid hormones, and these are going to be derived from that hydrocarbon-based cholesterol, or cholesterol, as my organic professor used to always say. And these are going to be very hydrophobic. These are not going to play well with water. They bind to receptors in the cytosol or of the cell's nucleus to generate their effects. Now, how do hormones travel through the blood? Well, either freely or they may be bound to proteins in the plasma. Free hormones are generally going to be those small amino acid-based hormones that are hydrophilic, able to freely associate through the water-based plasma of the blood. Note that there are a few notable hydrophilic hormones like growth hormone that are produced by the anterior pituitary gland that are also protein bound. Speaking of protein bound hormones are going to form complexes with binding proteins in the plasma. These are generally hydrophobic and do not associate well with the water molecules in the plasma. Now, why bind the proteins at all? Well, first, it allows the hydrophobic hormones to be each safely transported through the watery environment of the blood. It also gives the body a reservoir of hormones that can be released as the need arises. This can help prevent the concentration of free hormones in the plasma from experiencing any large fluctuations. And finally, it'll extend the lifespan of a hormone in the blood, which later we'll talk about as half-life. Now, as I said earlier, hormones really only interact with their target cells that contain proteins referred to as receptors. And these receptors are where those hormones bind. Receptors generally have a three-dimensional shape that is very highly specific for their hormone and can bind to hormones present at extremely low concentrations. And as I said, these hormones receptors are either embedded in the plasma membrane or they may reside within the cytosol or the nucleus of a target cell. Location of that receptor is generally dependent on whether the hormone is hydrophilic or hydrophobic. Since hydrophilic hormones cannot readily cross the phospholipid bilayer of the plasma membrane, that's where those hydrophilic receptors generally are located. By contrast, hydrophobic hormones can easily cross those membranes because of the interaction between that hormone and the membrane's fatty acid tails. It's able to just pass right on through, as shown in this photo. Now, what kind of hormone can cross the plasma membrane and interact with receptors in the cytosol or the nucleus? Which one goes into the cytosol or nucleus? It's a steroid hormone. Very good. Those hydrophobic, hydrocarbon-based steroid hormones are the ones that go easily through the plasma membrane binding to something in the cytosol or the nucleus. Now, the number of receptors in the target cell really varies depending on the body's need. Certain conditions can cause a cell to produce more receptors. For example, when a particular hormone level in the blood declines, target cells may make more receptors for that hormone, thereby increasing that cell's sensitivity to the hormone. This is a process called upregulation. Do note that in some cases, this response of upregulation is in response to a temporary increase in the circulating hormone level that's able to enhance the hormone's effect on the target cells. However, prolonged exposure to a high level of a hormone can cause the opposite effect. Target cells decrease the number of receptors, and that is a process known as downregulation. And these mechanisms allow cells to have tight control over how they interact with those hormones. Now, several types of mechanisms can be set in motion when a hydrophilic amino acid-based hormone binds with the cell surface receptor of its target cell. One such mechanism is a second messenger system. In these systems, this is when the hormone is going to bind to its receptor, and this is going to activate one or more proteins associated with the plasma membrane, beginning a cascade of enzyme catalyzed reactions. Generally, this is a sort of end result of this forming a compound inside the target cell called a second messenger. In this case, the hormone is going to be the first messenger. And most second messenger systems operate through a group of proteins associated with those receptors called G proteins. And when a hormone binds to a receptor linked to a G protein, the G protein changes shape and disassociates into two subunits. Generally, one of those subunits then interacts with another protein, usually an enzyme, and this G protein subunit may either activate or inhibit the enzyme, depending on which G protein has been activated. If the enzyme is activated, it catalyzes the formation of a second messenger, which then initiates a series of changes in the cell, leading to some change in activity. Generally, a key feature of second messenger systems is that they allow for signal amplification. Basically, this means that one hormone is able to lead to hundreds of second messenger molecules. And the second messenger molecules then in turn activate or inhibit thousands of other chemicals downstream, then changing the events in that cell. A well-studied example of G protein's second messenger system is adenylate cyclase, or CAMP system. In this signal cascade, G proteins activate an enzyme called adenylate cyclase, which then catalyzes the formation of second messenger cyclic adenosine monophosphate, or CAMP. CAMP then changes the cell, including an activation of protein kinases, catalyzing the transfer of phosphate groups from ATP to another molecule in a process known as phosphorylation. In depth, this whole process is done with the hydrophilic hormone binding to the receptor, causing that G protein to split into two subunits. The G protein subunit activates the adenylate cyclase. Adenylate cyclase catalyzes the formation of CAMP, which is the second messenger. CAMP activates protein kinase A, with protein kinase A then phosphorylating specific proteins such as ATP. And so this example shows that adenylate cyclase CAMP second messenger system really working in its entirety. So first we have that hormone traveling through the ECF. It binds to the receptor site of the hormone receptor. The hormone receptor then activates, disassociating this G protein. The G protein travels, activating adenylate cyclase. Adenylate cyclase then catalyzes the formation of CAMP, which acts as the second messenger. CAMP then activates protein kinase A, which is right here. So now this is activated, and that allows protein kinase A to phosphorylate very specific proteins, adding that phosphorus to them. So you can see where one hormone binding here has this massive effect, this amplifying effect. And that's what we're seeing with these hormones. Now, as I said earlier, hydrophobic hormones, like steroids and thyroid hormone, are able to diffuse across the plasma membrane and enter the cytosol of their target cells. This allows them to interact with intracellular receptors, either located in the cytosol or in the nucleus. Generally, when a hydrophobic hormone binds to an intracellular receptor, this is going to form a hormone receptor complex, which then binds to specific regions of the DNA known as the hormone response elements. This is an action that generally changes the rate of synthesis of one or more proteins. So first we have our hydrophobic hormones that diffuse straight through that phospholipid bilayer straight into the cell. Then they're going to find their intracellular receptor and bind to it, which then, through a long process of import-export, I'm not going to get into, it is then imported. Just an easy way to say it is just imported into the cellular nucleus, where it will then bind directly to the DNA, and that is going to upregulate the production or downregulate the production of specific proteins. Really, technically, it upregulates or downregulates the production of mRNA, which then in turn produces or doesn't produce proteins. Now, either cell surface or intracellular receptor activation can trigger different mechanisms inside the cell, leading to several effects, including stimulating the secretion from an endocrine or exocrine cell, activating or inhibiting enzymes, stimulating or inhibiting mitosis or meiosis, opening or closing ion channels in the cell's membrane, altering its membrane potential, or activating or inhibiting transcription of genes that code for RNA or proteins, also known as gene expression. Now, the maintenance of homeostasis isn't just one hormone. It's actually the interaction of multiple hormones that may have complementary actions, meaning that they affect different target cells to accomplish a common goal. Hormones of the adrenal gland and the pancreas work together, maintaining homeostasis during exercise. Other hormones may act on the same target cell to exert the same effect. This is a process known as synergism, and so these hormones are synergists, and they have pronounced effects when acting together, more pronounced effects when acting together than when they act alone. Sometimes, however, hormones that act on the same target cell have opposite effects, and this is an antagonist effect, where they act on the same cell, but they work to inhibit each other's effects. So a common question is, how long do hormones circulate? If they're long-lasting, how long is that lasting? Well, the truth is they continue to circulate in the blood until they're either taken up by their target cell and latching onto a receptor, or until they're broken down and deactivated. The amount of time of any particular hormone really depends on both the amount of the hormone that is secreted by the endocrine gland and at the rate at which that hormone is removed from the blood. Some hormones are inactivated very rapidly in their target cells, typically by enzyme catalyzed reactions, but most are actually pretty slowly removed from circulation, typically by the kidneys, where they're eliminated in urine, or in the liver, where they're broken down in more enzyme catalyzed reactions. And the rate really depends on the structure and the degree to which they're bound to those proteins. When we discuss a hormone's rate of elimination, we're talking about its half-life. The amount of time it takes for the plasma concentration of that hormone to reduce by 50%. Generally speaking, hydrophobic hormones have the longest half-lives, typically often a week or more, due to that extensive protein binding. And then in contrast, hydrophilic proteins have pretty short half-lives, with even the smallest hydrophilic hormones having half-lives of only a few minutes to a few seconds even. So let's talk about the regulation of secretion of these hormones. It's either initiated by hormonal, humoral, or neural stimulus. With a hormonal stimulus, you're going to find that some endocrine cells will either increase or decrease their secretion, typically in a response to some other hormone interacting with them. Examples here like the hypothalamus, it regulates secretion from the anterior pituitary gland through secretion of releasing and inhibiting hormones. So we're going to see the example of two hypothalamic hormones, growth hormone releasing and somatostatin. Each of these will stimulate and inhibit secretion of growth hormone from the anterior pituitary gland, respectively. And again, we'll look at that in just a second. You'll also have humoral stimulus. Many endocrine cells will either respond to some sort of ion or compound in the blood as far as concentration. A concentration of either an ion or a chemical compound, like glucose or calcium ions. These stimuli are called humoral stimuli, typically reflecting the historical name for body fluids. If you've ever heard of someone having, someone's humors, or there's a disease that they have a disease with their humors, that's the old medicine term for body fluids. In another figure, we'll look at a cell in the pancreas releasing a hormone called insulin, and insulin is one of those that responds to an increased level of glucose in the blood. Then you also have neural stimuli, and this is just when some of these endocrine cells respond to signals from the nervous system. We've already talked about one of these. The adrenal medulla is stimulated by those sympathetic neurons during your fight or flight response, and so those neurons release just a little bit of epinephrine or norepinephrine into the adrenal medulla, and then that is going to stimulate the adrenal medulla to release more neurohormones, epinephrine and norepinephrine. And so that's a little bit of that touching back that we've already talked about. And so here we see that hormonal stimulus from growth hormone releasing hormone. This is GHRH, growth hormone releasing hormone, stimulating the secretion of growth hormone from that anterior pituitary gland. So you see that the anterior pituitary receives growth hormone releasing hormone, and then it will then release growth hormone into the blood. And that's just a response to a hormonal stimulus. And that's the hormonal stimulation. Then you also have hormonal inhibition, where you see somatostatin binding to those same cells through different receptor sites. But what that does is it inhibits the release of growth hormone. So that is other endocrine glands producing an effect on these endocrine glands. And we'll talk about the specific term for that in just a little bit, but a teaser here, that's called a tropic effect. And here you have the other two types of stimuli that we talked about. You have the humoral stimulus, like shown here in the pancreas, where there's an excess of glucose in the bloodstream, and this pancreatic cell detects that, and so it releases the insulin responsible for converting that into glycogen. You also have those neural stimulus. So this would be the axon of one of those sympathetic pathways, and it releases those neurotransmitters into the adrenal medulla, and then the adrenal medulla releases neurohormones, epinephrine and norepinephrine, into the bloodstream to carry out those long-lasting effects. And we've already discussed this in our SNS chapter. Let's see if you remember. Which one of these has a longer half-life, hydrophobic or hydrophilic hormones? I want you to remember that half-life depends on the extensiveness of their protein binding. Hydrophobic hormones have longer half-lives. And remember, again, that is because they're more likely to be bound to those proteins, and that makes breaking them down harder, and so they last in the bloodstream longer. So hormone secretion is generally a negative feedback loop as far as regulation is concerned. I want you guys to recall the feedback loops principle that you guys learned in AMP1. And these negative feedback loops, especially of the endocrine system, follow a sort of general step that we're going to look at in this figure. Please note that in this example, we're going to use an endocrine cell as both a receptor and a control center. So the first thing is a stimulus. A regulated physiological variable is going to deviate from its normal range. And this is either an increase or a decrease from its normal range. In this figure, we see that it is a decrease. So then receptors of the endocrine cell are going to detect this deviation of the variable. And that's because these cells continuously monitor those regulated variables, like glucose, calcium ion concentration, and body temperature. Well, after the detection occurs, this stimulated control center, which is oftentimes it's the same endocrine cell, it's either going to increase or decrease its secretion of a particular hormone. And so in the endocrine system, you're going to see that the endocrine cell that detects a change in the variable from this normal range is generally also the control center. When the control center, in this case the endocrine cell, is stimulated, it changes its secretion of the appropriate hormone. And then you're going to have the effector or the response. And this is essentially the hormone triggering a response in its target cells that moves conditions toward the normal range. So this hormone secreted by the endocrine cell, the control center, enters the blood and reaches its target tissue or tissues, where it triggers those changes that return the variable to a normal state, the normal range. And you're also going to have homeostatic range. And then, so that's as the variable returns to its normal range, feedback to the control center decreases that effector response. So as, let's say, calcium concentration goes back to normal, those calcium promoting hormones will lessen their response. Less of them will be secreted. And so when those homeostatic conditions have been restored, the control center returns hormone secretion to the normal range by either decreasing or increasing it, and then this in turn stops the stimulation to the target cells, decreasing or ending the response. Now let's talk about the hypothalamus and the pituitary gland. We're going to start getting into some of these control mechanisms for the actual endocrine system. And I want you to recall that the hypothalamus of the brain is that small collection of nuclei with many important physiological functions, like regulation of hunger, thirst, fluid balance, body temperature, sleep-wake cycle, as well as certain reproductive functions. And so the hypothalamus works closely with the pituitary gland, sharing that close anatomical as well as the physiological relationship. Several hypothalamic and anterior pituitary hormones directly control certain body functions, whereas there are others called tropic hormones, meaning that they control hormone secretion from other endocrine glands. Do be careful not to confuse tropic with trophic. Trophic hormones induce a growth of its target cell. And then I do want you to note that some tropic hormones are also trophic hormones. Now, some of the functions of the hypothalamus are performed directly by hypothalamic neurons, whereas others are accomplished by hormones targeted to the anterior pituitary gland. In this section, we're going to sort of examine this relationship by discussing the cells, hormones, and target tissues of the hypothalamus and the anterior and posterior pituitary glands. We're going to look at these a lot as we move through this chapter, so it is important to explore their role in regulating other endocrine organs. So here you're going to see the hypothalamus is that small antero-inferior portion of the diencephalon of the brain. It is connected to the pituitary gland, which is often called the hypophysis, by a stalk called the infundibulum. The pituitary gland is a really small organ, about the size of a bean, that sits in the sella turcica of the sphenoid bone. And as this figure shows, the pituitary gland itself is made up of two structurally and functionally distinct components. There's the anterior pituitary gland and the posterior pituitary gland. Each of these are called adenohypophysis and neurohypophysis, respectively. The differences in the two components are inherent in their names. Adeno in the adenohypophysis means gland, and so that means that this anterior pituitary is a true gland composed of that glandular epithelium, which secretes hormones. The neuro in neurohypophysis refers to the fact that the posterior pituitary gland is essentially made up of nervous tissue. And this shows the infundibulum, that stalk that connects the hypothalamus to the pituitary gland itself. I hope that I've made a thorough mess of this image and you can still understand it. Now, the hypothalamus and the pituitary gland have a specialized blood supply that allows them to deliver their hormones. Capillaries converge in the hypothalamus to form larger blood vessels called the portal veins. And these portal veins travel through the infundibulum. And these veins lead to a second group of capillaries in the anterior pituitary gland. And then systems like this, in which capillaries are drained by veins that lead to another set of capillaries, these are all collectively called portal systems. And so accordingly, this group of vessels is called the hypothalamic hypophyseal portal system. Oh, I almost made it through it. Let me try again. Hypothalamic hypophyseal portal system. Got it on that one. Now, really quickly, which of these is that neurohypophysis? Made up of mostly neural tissue. It is the posterior pituitary. Neurohypophysis is in the back. Adenohypophysis is in the front. And because the posterior pituitary is made up of mostly neural tissue, it makes no hormones of its own. Instead, what it does is it stores and releases two neurohormones produced by clusters of cell bodies in the hypothalamus, known as supraoptic and paraventricular nuclei. Now, these hormones are antidiuretic hormone and oxytocin, and we're going to look more closely at these hormones and their roles here. So the first one we're going to talk about is antidiuretic hormone, or ADH, or vasopressin. They're all the same thing. This is a hypothalamic hormone that controls water balance, and its synthesis and release process is basically this. First, those hypothalamic neurons are going to create ADH, and this is done in the cell bodies of hypothalamic neurons in the supraoptic and the paraventricular nuclei. These are going to continually make ADH in low amounts. Then the axons of the hypothalamic neurons are going to travel through the infundibulum as a group known collectively as the hypothalamic hypophyseal tract. And then ADH is going to be stored. So the axons of the hypothalamic neurons terminate in this posterior pituitary, where the ADH is then stored in synaptic vesicles. Finally, normally, an axon terminal forms a synapse with another cell. However, in the posterior pituitary, the axon terminal simply releases these hormones through exocytosis into that interstitial fluid, and eventually they will diffuse into local capillaries. This is normally stimulated by those action potentials of those axons. Now, the primary function of ADH is to increase the amount of water retained by these kidneys. This function is really apparent from its name. A diuretic is a chemical that increases urine production, which decreases the amount of water in the body. And so an antidiuretic has the opposite effect. It decreases urine production by increasing the amount of water retained by the body. So you can understand ADH a little bit better, we're going to look at a kidney diagram here briefly. Note that the fluid that eventually becomes urine is located within small tubules in the kidney, and the relationship between the tubules, the interstitial fluid, and the blood is going to be shown there. But first, ADH is going to increase that amount of water in the body by causing the insertion of water channels called aquaporins into the plasma membranes of the cells, forming those kidney tubules. Water can enter these cells only via aquaporins, and so aquaporins are inserted only in the presence of ADH. Aquaporins allow water in the tubules to enter the cytosol of kidney cells through osmosis, reducing the amount of water remaining in those kidney tubules. From the cytosol, water then moves into the interstitial fluid and then into the blood and the capillaries, again by osmosis. So the net effect of ADH is to return water to the blood that would have otherwise been eliminated from the body as urine. This is mostly regulated through osmoreceptors in the hypothalamus, monitoring changes in the solute concentration of the blood. These osmoreceptors respond by increasing solute concentration by stimulating ADH release from that posterior pituitary gland. This will lead to water retention, decreasing the solute concentration of the blood. Keep in mind that an increased amount of water dilutes the blood, which causes the relative concentration of solutes to decrease. Conversely, these same receptors respond abnormally to low solute concentration by inhibiting that ADH release, which leads to water loss and an increased solute concentration in the blood. An abnormal lack of ADH secretion or activity can result in a disease called diabetes insipidus. Now, signs and symptoms of diabetes insipidus include extreme thirst, dehydration, and a very high solute concentration in the blood, because this person's body is unable to conserve most of the water that is consumed. Most forms of diabetes insipidus are treated the same way. It's an administration of synthetic ADH. And please don't confuse diabetes insipidus with diabetes mellitus. This is a separate condition that we'll discuss in a little bit. Diabetes simply refers to the term of excessive urination. So here's that kidney figure that I was talking about. And we're going to see a zoomed in version of the kidney here. And we're going to see this is the tubule. This whole yellow structure is the tubule. And those are this lumen inside is full of the fluid that would normally become urine. Now, in the presence of ADH, those aquaporins are going to open up in these cells, allowing water to pass through from this fluid in the tubule out into the interstitial fluid here, and then into the capillaries eventually here. So that's just pulling water out of what would be the urine and then back into the blood. It's a process of resorption, basically. Now, much like ADH, oxytocin is produced by the hypothalamus and stored in the axon terminals of the posterior pituitary gland. Its functions, however, are different. Oxytocin's functions are primarily reproductive in nature, targeting specialized cells of the mammary glands and smooth muscle cells of the uterus. In nursing mothers, oxytocin release is triggered by an infant suckling. And this oxytocin binds to those specialized cells that stimulate their contraction, resulting in milk ejection. This is an action known as the milk letdown reflex. It's a classic example of a positive feedback system to the body, as lactation induces the hungry infant to suckle more vigorously, stimulating more milk release. This positive feedback loop is going to be broken when the infant becomes full and no longer suckles, though. This is another example of that feedback loop's core principle that you guys should understand by now. And before I talk about this photo, I want to say that oxytocin also has neural effects as well. The possible role of oxytocin in promoting emotional bonding in both sexes is currently under investigation, but it is thought to be the sort of quote-unquote love chemical. These topics, including how oxytocin functions in childbirth, are going to be more closely covered in the development and heredity chapter when we get there. But this figure shows essentially what we mean when we say that these hypothalamic hormones are stored in the posterior neurohypophysis, this posterior pituitary. They're created up here in those supraoptic and paraventricular nuclei, and then they travel down the hypothalamic hypophyseal tract, and then they just sort of wait in the axon terminals in their synaptic vesicles. Then when there's a triggering event, they'll be secreted into the capillaries there, which then circulates them throughout the body to their target cells. So hypothalamic hypothalamic hormone created in the hypothalamic neurons, which travel down and just wait in the posterior pituitary. Now, what is the result of antidiuretic hormone? Which one is antidiuretic hormone? So this means you need to know what diuretic hormone or what a diuretic does, and then what is the opposite of that? It is decreased urination with increased water retention. Remember, diuretics increase, this would be a diuretic hormone right here, increasing urination and decreasing water retention. Now, the hypothalamus is going to control many of the anterior pituitary gland's functions through the production and release of tropic hormones that are going to affect how the anterior pituitary gland secretes its own hormones. These hormones from the hypothalamus are called releasing and inhibiting hormones because they either stimulate or inhibit the release of hormones from the anterior pituitary. The hypothalamic releasing and inhibiting hormones are usually delivered via the hypothalamic hypophyseal portal system via secretion into that hypothalamic capillary bed, where they then travel through portal veins in the infundibulum, exiting the inferior pituitary capillary bed, binding onto receptors on the anterior pituitary cells that then stimulate or inhibit the secretion of hormones from the anterior pituitary cells. But most hormones of the anterior pituitary gland itself are actually primarily tropic. They control the secretion of hormones by other endocrine glands or tissues, with one notable exception. Growth hormone is more widespread, is an anterior pituitary gland with more widespread actions. Now, the hormones of the hypothalamus, the adenohypophysis, as well as hormones secreted by the anterior pituitary's target tissues are maintained at normal levels, usually by negative feedback loops of multiple tiers of control. Each tier is then going to have a stimulus, receptor, control center, and a response or effector. So these are very critical for maintaining those homeostatic levels. So the first tier of feedback control really involves neuroendocrine cells of the hypothalamus. And this is going to then secrete the releasing or inhibiting hormones, which then stimulate or inhibit hormone production from the anterior pituitary, which is the second tier of control. The pituitary hormones then stimulate cells of their target organs. Many of them are other glands, which then act as the third tier of feedback control. The glands of the target organs secrete hormones that affect some well-regulated physiological variable there. When the variable is returned to a normal range, typically around a set point, secretion of releasing hormones from the hypothalamus decreases, and or secretion of inhibiting hormones increases. And so as a result of these changes, secretions from the anterior pituitary gland will decrease, and secretions from the endocrine target cells will in turn decrease. So it's just a sort of cascading effect. These multiple levels of control allow that endocrine system to tightly regulate conditions within the body, which then is critical for maintaining the homeostasis. And this is that multi-tiered negative feedback control system that we talked about with the hypothalamus, the anterior pituitary gland, and then their target organs or cells. So first tier is hypothalamus, second tier is anterior pituitary, third tier being target organs themselves. And each of these then has their own sort of hormones that they produce. Typically, these are either going to be synergistic or antagonistic. But the first tier is, that those releasing hormones, which will then either inhibit or promote the secretion of anterior pituitary hormones, which then go to the target cells, and then they either inhibit or promote the effects that they're looking for to maintain that homeostasis. Now, as I said, the anterior pituitary gland secretes many hormones, most of which are tropic, like the thyroid stimulating hormone. As implied by its name, thyroid stimulating hormone, also known as TSH, also known as thyrotropin. This stimulates development of the thyroid gland and its secretion of thyroid hormones. TSH is produced and secreted by cells called thyrotrophs, and its secretion is triggered by a hypothalamic releasing hormone known as thyrotropin releasing hormone, TRH. The secretion of TSH is inhibited by the hypothalamic hormone somatostatin. Somato basically means block, and remember, somato means body, and statin means block. So this is just sort of inhibiting the body there. Then you also have adenocorticotropic hormone, or otherwise known as corticotropin, and it is produced by cells called corticotrophs. It stimulates the development of the adrenal glands, and then there's synthesis of various steroid hormones. ACTH secretion is stimulated by a hypothalamic releasing hormone known as corticotropin releasing hormone, CRH. You've also got the hormone prolactin, which is produced by cells called lactotrophs. And it has several target tissues in both males and females where its effects are tropic, but its primary target tissue is the mammary gland cells, where its effects are non-tropic. Here, it is secreted generally in women who have given birth, where it then stimulates the growth of mammary glands and the initiation of milk production after childbirth. And then it also maintains milk production for the duration of breastfeeding. The main stimulus for prolactin release is the suckling of the nipple by an infant. And normally prolactin secretion is inhibited by the hypothalamic inhibiting neuron. The hypothalamic inhibiting hormone, prolactin inhibiting factor, now known to actually be dopamine. Suckling by the infant decreases the release of dopamine, which facilitates more prolactin release. You also have luteinizing hormone, which is one of two hormones known as gonadotropins. These are secreted by cells called gonadotrophs. The gonadotropins are named because what do? They stimulate male and female gonads, the testes and the ovaries respectively. So luteinizing hormone secretion is stimulated by the hypothalamic releasing hormone called, you guessed it, gonadotropin releasing hormone. In males, luteinizing hormone is going to stimulate production of the male hormone testosterone by cells of the testes, and then in females it produces female hormones called estrogens and progesterone, and it also triggers the release of an oocyte, an egg cell, in the process of ovulation. You've also got follicle stimulating hormone, which is the other gonadotropin. Like luteinizing hormone, FSH is stimulated by gonadotropin releasing hormone from the hypothalamus. And in males, follicle stimulating hormone stimulates cells of the testes to produce chemicals that work to bind and concentrate testosterone, where in females, follicle stimulating hormone works with luteinizing hormone to trigger the production of estrogens. And it gets its name from the fact that it also triggers the maturation of ovarian follicles, which then house those developing oocytes. And we're going to discuss TSH and ACTH in more detail in the upcoming chapters. And then luteinizing hormone and follicle stimulating hormone are going to be also explored a little bit further. And then also more in depth when we discuss the reproductive and development, reproductive system as well as development chapters. And so this is that path for the anterior hormone. The anterior posterior, the anterior pituitary hormone production. You're going to see that the hypothalamic neurons secrete those releasing hormones into this, the hypothalamic hypophyseal portal system. And then they are carried down into the anterior pituitary, where they will then bind to their target cells and either stimulate or inhibit the secretion of hormones from these anterior pituitary cells. Now, just really quick, which of these is actually secreted by the hypothalamus? Which of these is secreted by the hypothalamus? It's thyrotropin releasing hormone. Remember, the hypothalamus only releases releasing hormones. And then which of these is released by the anterior pituitary? Anterior pituitary releases which of these? Luteinizing hormone. Gonadotropin here. Gonadotrope releasing hormone from the hypothalamus. These are produced by the ovaries, and this is produced by the testes. So luteinizing hormone is the only one actually produced by the anterior pituitary. Now we're going to talk about some hormones produced by the anterior pituitary that have a little bit more widespread effects. The first of which is growth hormone. Growth hormone is produced and secreted by cells called somatotrophs, and growth hormone itself is sometimes called somatotropin. These cells that release growth hormone periodically will release it throughout the day, and actually the peak secretion occurs during sleep. And its function is pretty apparent by its name. It regulates and controls growth. The effects on growth hormone on target tissues, including skeletal and cardiac muscle, adipose tissue, liver tissue, cartilage, bone. These effects can either be short-term or long-term. Short-term effects of growth hormone are primarily metabolic in nature, typically promoting fat breakdown and the production of new glucose by the liver, this is a process called gluconeogenesis. And they also, another effect is the inhibition of glucose uptake by muscle fibers. These effects will increase the concentration of glucose and fatty acids in the blood, allowing cells to use them as fuel and raw materials for growth. In this photo, we can basically break it all down as far as short-term and long-term effects. The short-term effects you see, as it's released from the anterior pituitary, it goes through the blood vessels into its target cells. It can inhibit glucose uptake. It stimulates gluconeogenesis in the liver, stimulates lipolysis in fat, and each of these are going to either increase glucose concentration or increase blood fatty acid concentration. And essentially, that's good for creating those raw materials to be used by cells. Now, the long-term effects of growth hormone are not all mediated by growth hormone directly. Instead, what will happen is growth hormone will act on the liver and other target tissues to promote the production of a hormone called insulin-like growth factor. And its name reflects the fact that it functions very similarly to the hormone insulin, which we'll discuss later. IGF affects nearly every cell type in the body, where it will pretty much trigger rapid protein synthesis and cell division, leading to increased longitudinal bone growth, and as well as muscle development in children. It also decreases blood glucose concentration by stimulating glucose uptake by cells, which is really the opposite action of acute growth hormone release. And then both hormones continue to play important roles in adults, even after those longitudinal bone growth has ceased. And it promotes muscle development as well as regulating body mass. And these effects have really led to a sort of unsavory promotion of growth hormone as a quote-unquote health supplement. And we'll look at that when we look at the sort of the clinical A&P aspect of it. Growth hormone is really regulated by two hypothalamic hormones. The first is growth hormone releasing hormone, and GHRH is going to stimulate the release of growth hormone, in particular during exercise, during fasting, and during stress, and then after the ingestion of a really protein-rich meal, so like meats and soy protein shakes, something like that. Growth hormone release is inhibited by somatostatin, just like thyroid stimulating hormone, and it is that somatostatin is also known as growth hormone inhibiting hormone. So here you see that release and regulation of growth hormone, stimulated by the hypothalamus releasing GHRH, growth hormone releasing hormone, which travels to the anterior pituitary. Anterior pituitary releases growth hormone. Growth hormone goes, has its effects, and then eventually the anterior pituitary is going to decrease the release of growth hormone, typically by allowing the hypothalamus to secrete somatostatin, that growth hormone inhibiting hormone. So this is another negative feedback loop where, okay, we've reached homeostasis, so now we need to stop, and so now they'll just release a new hormone to stop production. Now, there are some imbalances in growth hormone secretion that can cause several disorders. Generally, hypersecretion of growth hormone may be caused by a tumor. Has two different diagnoses coming out of it. If this hypersecretion occurs before the closure of the epiphyseal plates, typically, during a person's puberty, this person is said to be diagnosed with gigantism. Individuals with gigantism are unusually tall, probably more than 7 feet. Not only does the skeleton increase in size, but all other tissues, including the heart and other organs, will enlarge as well. Generally, if this condition is detected early enough, it can be managed typically by removal of the tumor or the anterior pituitary as a whole. Um, However, if left untreated, affected individuals generally succumb to heart failure at an early age. The photo on the right is, of the upper right, is of Robert Wadlow, uh, medicine's tallest man at almost 9 feet. He died at 22 of a heart attack, simply because his heart had grown too large, and it was just unable to supply his body with blood effectively. If this hypersecretion occurs after the closure of epiphyseal plates, the result is a different condition. It's called acromegaly. Uh, Longitudinal growth can no longer take place, so tissues instead increase in girth, particularly those of the head, face, hands, and feet, as well as vital organs like the heart and liver. The lower right photo here is of famous actor slash French wrestler Andre the Giant, and he is said to have been diagnosed with acromegaly. If left untreated, of course, this will lead to disruptions of various organ systems up to and including headaches, high blood pressure, and heart failure. And so there's hyposecretion disorders as well. A person that undergoes hyposecretion of growth hormone before puberty is considered to be a pituitary dwarf with pituitary dwarfism. People with this condition are generally short in stature, but their limbs and their trunk are proportional. On the left hand here, you see an individual with pituitary dwarfism. A similar form of this dwarfism will result when IGF, insulin-like growth factor secretion, is abnormally low, although these patients have normal to elevated growth hormone secretion levels. So this table actually gives a summary of all of the hypothalamic hormones that are going to be stored in and released from the posterior pituitary. And then it also gives it detail on regulatory hormones of the hypothalamus as well as hormones of the anterior pituitary. And I'm not going to go through all these again. I've already covered all of them, but I will just go through these. I'll give you a couple seconds on each slide to pause it and study this table as you need to. So I'm sure everybody knows of that myth of, oh, human growth hormone, HGH, it promotes fat loss and muscle growth, and it's literally the fountain of youth. I mean, that's what people say. It promotes fat loss and muscle growth and is a amount of youth, but it is a popular supplement with bodybuilders and people that are seeking weight loss and individuals that are looking to stave off the inevitable effects of aging. And there is a grain of truth to these assertions. Growth hormone does indeed promote fat breakdown and protein synthesis, and patients with growth hormone deficiency are treated with HGH. They do lose fat and gain muscle. However, serious problems arise with HGH supplementation. Excessive GH can cause many potentially harmful effects, like elevated blood glucose and growth of organs such as the tongue and bones of the hands, feet, and face. However, most HGH supplements fortunately come with very few risks because they're sold as oral preparations to be ingested. You might be thinking, well, why does like a pill mean that the risk is lowered? Well, that's because growth hormone is a protein hormone, and so it is really destroyed by acid and enzymatic reactions in the stomach. The only really effective way to administer this drug is via injection, and so oral preparations like tablets or pills really do little to really amount to little more than expensive feces. Uh, Someone taking such supplements should still be careful, though, as they can contain ingredients that may be untested for safety or efficacy. There's no cheap and easy way out, and pills are a waste of time as far as growth hormone is concerned. So test yourself really quick. Target cells make more receptors for a particular hormone when the level of that hormone declines in the bloodstream. This process is called what? The process of a target cell making more receptors for a particular hormone, while that hormone declines concentration in the bloodstream. That is upregulation, making those cells more sensitive to the lower concentration of that hormone in the bloodstream. Increasing blood glucose stimulates the release of insulin. This type of stimulation is called which kind? Increasing blood glucose stimulates the release of insulin. This type of stimulation is called humoral stimulation. Remember, this is a reaction to solutes like ions or solutes in the blood creating an insulin release. And where is antidiuretic hormone, also known as ADH or vasopressin, where is that made? Is it made in the anterior pituitary, posterior pituitary, hypothalamus, or thyroid gland? It's the hypothalamus. It's made in the hypothalamus, and then it is stored in the posterior pituitary, where it is then secreted into the bloodstream. But remember, the neurohypophysis, the posterior pituitary neurohypophysis, does not make any of its own hormones. It only stores and secretes them. Now let's talk about the structure of the thyroid and the parathyroid glands. So the thyroid gland is a butterfly-shaped gland consisting of right and left lobes connected by a small isthmus, this band in the middle. It looks sort of like a bow tie or a butterfly, and microscopically, this thyroid gland is composed of multiple spheres known as thyroid follicles. And here we see the thyroid itself. The interior of one of these follicles is filled with colloid, a protein-rich gelatinous material, which is where the precursor of thyroid hormones is stored. Colloid also contains a high concentration of iodine atoms, which are required for thyroid hormone synthesis. And between the thyroid follicles lie clusters of parafollicular cells. Now, these cells are larger than follicle cells, and they have no role in any hormone production, any thyroid hormone production. Instead, these are going to produce the hormone calcitonin, which we're going to discuss a little bit later. So this is actually an artist's rendition of those follicle spheres, okay? This is a thyroid follicle containing colloid, and this would be one of those follicles. And then inside of it, you're going to see the surrounding follicular cells, and then this fluid, this colloid precursor for that thyroid hormone. And then here you have the actual histology, where you see the surrounding follicular cells all around that big empty lumen, or big, I guess it is full lumen, full of that colloid material. Now, which hormone is made by the parafollicular cells of the thyroid gland? T4, T3, parathyroid hormone, or calcitonin? It is calcitonin. Now, the parathyroid glands are very small glands that are embedded in the posterior surface of the thyroid gland itself. Typically, there can be between 3 and 5, with 4 being the most common. That bilateral symmetry thing. We really want 2 on one side and 2 on the other. This hormone-secreting cells of the parathyroid gland commonly known as chief cells, they're going to produce a hormone called parathyroid hormone. Now, thyroid hormones consist of an amino acid core that is bound to iodine atoms. And there are two forms of thyroid hormone, typically named according to the number of iodine atoms they have. There's triiodothyronine, okay? Triiodothyronine, otherwise known as T3, which contains 3 iodine atoms. And then there's thyroxine, T4, which has 4 iodine atoms. Now, both forms of this hormone are active, but T3 has a far greater physiological activity than T4. And in fact, as we're going to discuss in a little bit, T4 is usually converted into T3 in those target cells. Now, as I said, they are amino acid based, but their amino acids are nonpolar, and so they are hydrophobic, and they do not bind well to cell surface receptor proteins. Instead, they diffuse into the target cells, and then they bind to intracellular receptors of the nucleus. Here, just like steroid hormones, they either activate or inhibit the transcription of certain genes. So this section is really going to cover the functions of thyroid hormones, and then we're going to see how they're produced, how that production is regulated, and what happens when this production is imbalanced. Now, nearly every cell in the body contains receptors for thyroid hormones, so their effects are pretty widespread. Broadly, there are 3 categories. There's the regulation of metabolic rate and thermoregulation. There's the promotion of growth and development, and there's synergism with the sympathetic nervous system. The first one is regulation of the metabolic rate and thermoregulation. Now, thyroid hormones are going to be what sets the basal metabolic rate, and this is basically the minimum amount of energy required by the body to carry out all of its resting reactions. And the hormones accomplish this in several ways. Increase ATP synthesis for sodium-potassium pumps, which then raises the amount of ATP that the cell consumes. The thyroid hormones can also act on a number of different cell types to trigger other energy-requiring reactions, like acting on cells of the liver, triggering gluconeogenesis. They can also stimulate reactions that break down proteins in skeletal muscle and fats and adipose tissue in order to provide these compounds for gluconeogenesis. And then finally, thyroid hormones can also initiate energy-requiring reactions in these same tissues that build proteins and fats. So thyroid hormones will increase the rate at which cells carry out both catabolic, breaking down, and anabolic, building up, reactions, essentially leading to no net change in the overall cell's composition. Now, why expend energy in a way that leads to no net change? Well, the answer lies in the byproduct of these reactions. It's heat. And so if you guys remember that energy transfer, as far as our cellular reactions go, is very inefficient. And so much of the energy that we create is actually lost as heat. The heat from these reactions helps maintain our core body temperature in a process called thermoregulation. So although these actions of thyroid hormones may seem quote-unquote wasteful, they're actually crucial for maintaining that internal body temperature. Thyroid hormones are also required for normal bone growth, normal muscle growth, and nervous system development. Now, the exact roles are not really understood fully as far as thyroid hormone is concerned, but thyroid hormone deficiencies definitely result in stunted growth and problems with nervous system development, as we'll discuss in a little bit. So there is a very close link, but it's not fully understood. And elevations in thyroid hormone also act on target cells of the sympathetic nervous system that can help upregulate receptors for sympathetic neurotransmitters. In this way, the thyroid hormones kind of help regulate blood pressure, heart rate, and other sympathetic nervous system actions without directly causing these effects. Now, the multi-step synthesis of thyroid hormone occurs both in the colloid and in the follicle cells. First, the synthesis begins with an accumulation of iodide ions. These are I negative ions in the colloid. These iodine ions are ingested by food, via food, after which they're absorbed into the blood. In the capillaries of the thyroid gland, these iodide ions diffuse into the interstitial fluid and are actively pumped into the follicle cells. Finally, the ions then enter the colloid because transport proteins in the plasma membranes of the follicle cells will grab this iodide and then force it in there. In addition, follicle cells secrete the protein thyroglobulin, and this is going into the colloid through exocytosis. Thyroglobulin is a very large protein, typically about 100 residues of the amino acid tyrosine. Now, thyroid hormones are typically made from these tyrosine residues. So after the iodide is secreted into the colloid, iodide ions have to be converted from iodide into iodine atoms. And then these iodine atoms will then get attached to the thyroglobulin. And this is done through an enzymatic reaction, converting iodide to iodine. Iodine ions, or I'm sorry, they're no longer ions, they're atoms. Iodide ions get converted into iodine atoms, which then move into the colloid and attach to those selected tyrosine residues on the thyroglobulin, forming a structure called iodinated thyroglobulin. And then some of the tyrosine residues receive only one iodine atom, forming monoiodothyronine, or MIT, whereas others receive two iodine atoms, forming diiodothyronine, or DIT. Now, these iodinated thyroglobulin needs to enter the follicle cell through endocytosis, and then T3 and T4 are cleaved from the molecule in reactions that are catalyzed by lysosomal enzymes. The iodinated thyroglobulin is taken up by the follicle cells, where the endocytic vesicles will fuse with lysosomes, where the enzymatic reactions will then cleave the MIT and the DIT from the thyroglobulin. If an MIT molecule is combined with a DIT molecule, the result is T3. If two DIT molecules are combined, the result is T4. And now about 90% of the hormones produced are T4, while the remainder is T3. And T3 and T4 are then released into the blood, where the vast majority of both will bind to plasma proteins. Any unbound hormones are known as free T3 and T4, while only free T3 and T4 have any biological activity. Keep in mind that, yes, those target tissues will convert that T4 to the T3, but the main reason T4 is produced in such massive quantities is that T4 provides that T4 provides the cells with a much longer half-life of about 7 days, where T3's is only about 24 hours. And so those cells' ability to convert that T4 into T3 really makes it much more simple to just mass produce T4, have it stick around longer, and then convert it when it's needed. And so here you see those iodide ions that will need to travel from the interstitial fluid from the blood vessel into the interstitial fluid, and from the interstitial fluid into the follicle cell through endocytosis. Eventually, the iodide ions and the thyroglobulin, here you see in a vesicle, all of them get pushed into the colloid from the follicle cells. Iodide ions then get converted into iodine atoms, which then attach to the thyroglobulin, either MIT or DIT. You see an MIT here, and you see some MITs there and a DIT here. All of that then will get become known as iodinated thyroglobulin, which then enters the follicle cell through endocytosis, where it joins up with a lysosome. Okay, you see, which is indicated here, and the lysosome is full of those enzymes, which are then going to cleave off the now combined iodine into either T3 or T4. Or remember, 90% of it is T4. And then that T3 and T4 leave the follicle cell and enter the bloodstream. Now, as with most hormones, T3 and T4 production is regulated through that negative feedback loop. But it is important to note that this negative feedback loop has multiple tiers of control, involving both thyrotropin-releasing hormone from the hypothalamus as the first tier and thyroid-stimulating hormone from the anterior pituitary as second tier. Hypothalamic neurons are going to secrete that TRH at a fairly consistent rate, which then will travel to the anterior pituitary via the hypothalamic hypophyseal portal system, and it stimulates production and release of TSH. TSH stimulates multiple responses from the thyroid gland, including production of T3 and T4 by follicle cells, secretion of T3 and T4 from follicle cells into the blood, as well as growth and development of the thyroid gland itself. And the third tier production is really just generated by the third tier of control is generated by the thyroid itself. So production and secretion of TRH and TSH increase when levels of free T3 and T4 fall, or when the body is exposed to really cold temperatures. Secretion of both TRH and TSH is also inhibited by rising levels of T3 and T4. And so, as mentioned earlier, the hypothalamic hormone somatostatin also is going to inhibit this TSH production. And so here's that negative feedback loop where you see the first tier is what? It's the hypothalamus secreting TRH. Second tier is the anterior pituitary secreting TSH, which then stimulates the third tier control, which either produces T3 and T4, secretes T3 and T4 into the blood, or it grows the thyroid as the thyroid gland itself. And then the effects are increased levels of T3 and T4 in the blood and an increased metabolic rate. Now, if there's too much T3 and T4, the hypothalamus is going to decrease TRH secretion, which then decreases TSH production or secretion, which then doesn't stimulate the thyroid as much. So now it's not producing as much T3 and T4. And then when levels fall below a certain point again, it's going to kick up that TRH, which kicks off TSH, which kicks off T3 and T4 production. It's just this big long cycle, trying to stay in that homeostatic range. Now, due to the fact that systemic effects of thyroid hormones are just widespread, serious imbalances in homeostasis can accompany their over or underproduction. Overproduction of thyroid hormones is called hyperthyroidism. Underproduction is hypothyroidism. With the most common cause of hyperthyroidism being Graves' disease, which results from the immune system producing abnormal proteins that are going to mimic TSH on the thyroid gland. The abnormal proteins stimulate that thyroid, and the levels of both T3 and T4 in the blood increase. Common signs of Graves' disease result from excessive T3 and T4 activity and include weight loss, heat intolerance, and disruptions in heart rhythm and increase in blood pressure due to this synergism of thyroid hormones with the sympathetic nervous system. You're also going to find that patients of Graves' disease may also suffer from exophthalmos, and this is characteristically shown by the bulging of the eyeballs which is caused by weakened extrinsic eye muscles. And then they also have water retention in the tissues around the orbits, with a final sign of hyperthyroidism being the enlargement of the thyroid gland known as a goiter. And thyroid enlargement results from the abnormal TSH-like proteins secreted in Graves' disease, which stimulate that growth of the thyroid gland itself. And here you see a patient with what appears to be the right lobe of their thyroid engorged in the goiter, with a goiter, and here you see those bulging eyes of exophthalmos, and also you're going to see a little bit of fluid retention in the orbits surrounding them. But generally, these are two of the very common symptoms for Graves' disease. Typically, hyperthyroidism is treated with drugs that decreases the production, but these drugs are pretty much a lifetime solution. Permanent treatments can basically involve thyroidectomies or thyrectomies, which is just the surgical removal or the non-surgical destruction of the thyroid gland. This is typically non-surgically done when the patient takes radioactive iodine, which is selectively taken up by the follicle cells, and then the radiation actually produced as the iodine decays kills those follicle cells. This is also why nuclear fallout is so dangerous for human beings, because we'll so easily take up radioactive iodine, and the thyroid is one of the only structures in the human body that uses it, and because it has such systemic, widespread effects, cancer of the thyroid is can really produce some devastating effects. Now, the two most common causes of hypothyroidism are iodine deficiency and the destruction of the thyroid gland through immune system action, with that immune system condition actually being known as Hashimoto thyroiditis. And immune deficiency hypothyroidism is relatively uncommon in the first world, because most table salt nowadays is supplemented with iodine. And people who regularly consume food from the ocean, they're also likely to receive natural iodine source from their food. However, in some developing countries, iodine deficiency remains relatively common, with both causes of hypothyroidism resulting in decreased T3 and T4 production, and then in concurrent, this is going to increase TRH and TSH production. Remember, if there's not enough T3 and T4 in the blood, they're going to keep sending out that TRH and TSH. And most signs and symptoms of hypothyroidism are simply the opposite of those of hyperthyroidism. And those symptoms include a weight gain, cold intolerance, and a slow heart rate. But many patients with hypothyroidism do present with one of the same signs, a goiter. A goiter develops in Hashimoto thyroiditis because of the increased level of TSH promoting thyroid gland growth. In iodine deficiency hypothyroidism, an elevated TSH level will contribute to the goiter, but the goiter instead results from the accumulation of thyroglobulin in the colloid of the thyroid follicles. And so goiters due to iodine deficiency, they can actually become quite large. And so treatment of hypothyroidism really depends on the cause. Iodine deficiency typically resolves with iodine supplementation. Hashimoto thyroiditis has no cure, but it can be easily treated with thyroid hormone supplementation for the remainder of that patient's life. Now, there is a special variant of hypothyroidism called congenital hypothyroidism, formerly known as cretinism. And this develops when an infant is born with an inadequate thyroid function. If untreated, this is going to result in delayed physical and nervous development and then eventual intellectual disability. Congenital hypothyroidism is completely avoidable with an early diagnosis and treatment, and so for this reason, infants are screened for hypothyroidism at birth. So, thermoregulation is the responsibility of what hormone? What hormone is it that keeps that basal metabolic rate at neutral? Those are the thyroid hormones. Yes, very good. T3 and T4. As I briefly touched on in the introduction for this, calcitonin and parathyroid hormone are going to be produced by the thyroid and parathyroid glands respectively. Now let's take a closer look at the functions and the target tissues of these hormones, as well as their roles in maintaining calcium ion concentration in the blood within a specific range. So, the chief cells of the parathyroid gland are going to be what secretes the peptide hormone, parathyroid hormone, PTH. And this is a major factor in maintaining that blood calcium ion concentration, at least keeping it within normal range. PTH is secreted in response to a declining blood calcium ion concentration in a condition known as hypocalcemia. And this it has several actions to raise the calcium ion level of the blood through triggering various effects. So PTH induces changes in the osteoblasts that enhance osteoclast activity. I want you guys to remember that osteoclasts are bone-destroying cells that release calcium ions into the blood by breaking down the extracellular matrix of bone. As the osteoclasts degrade bone, calcium ions are released back into the blood. The effects of PTH on the small intestine are sort of indirect and involve the hormone vitamin D, which is produced by the skin in response to certain wavelengths of UV light. Now, however, the skin produces an inactive form of vitamin D. Parathyroid hormone is going to act on the kidneys to stimulate the conversion of vitamin D from inactive to active. Vitamin D3 is its active form, otherwise known as calcitriol. In this form, vitamin D3 increases the number of calcium ions absorbed from the small intestine. In the process of filtering blood, the kidneys also remove large amounts of water and solutes from the blood, including many calcium ions. The cells of the kidney tubules then reclaim most of the water and the solutes through a process called tubular resorption. PTH acts on these kidney tubule cells to increase the number of calcium ions reclaimed from that fluid in the tubules before it becomes urine and is lost to the body. So this is going to be the negative feedback loop involving parathyroid hormone that maintains the blood calcium ion concentration as normal range. So the first thing we have is a stimulus, and the stimulus is the blood calcium ion level dips below normal range. The receptor, the chief cells in the parathyroid gland, are going to notice a decrease in that calcium ion level in the blood because of their cell surface receptors. The chief cells, acting as the control center, are going to increase their production and secretion of parathyroid hormone into the blood. This is going to affect the function of its target cells in bone, in this case it's the osteoclasts, as well as the kidneys and the small intestine. In addition, it's going to stimulate that conversion from inactive vitamin D to active calcitriol. All of these work together to increase the concentration of calcium ions in the blood. And then as the calcium ion concentration in the blood returns to normal range, receptors on the chief cells are going to detect that change, and the negative feedback decreases their production and secretion of PTH, which then decreases the stimulation of the target cells, decreasing all of that calcium ion resorption or production. And so that response is now reduced. Now, calcitonin is secreted in response to a condition called hypercalcemia. And this is when the calcium ion level in the blood increases above normal range. Calcitonin, which remember is produced by the thyroid gland's parafollicular cells, this works to decrease the blood calcium ion concentration. And calcitonin's primary actions involve inhibiting the activity of the osteoclasts in the bone. And this leaves the effects of the osteoblasts, the bone building cells, unopposed. Osteoblasts are going to then be able to deposit calcium ions from the blood into the bone, reducing the amount of calcium ions floating free in the blood. And although calcitonin's effects are pretty difficult to characterize, mostly because they're fairly transient, the effects of a high calcitonin level appear to only last for a few hours. It is important to note that its activity is really important during times of active bone turnover, like bone growth. At present, calcitonin is not believed to be a major regulator of calcium ion homeostasis and may actually instead play a more important role in bone homeostasis. Both calcitonin and PTH are used to treat osteoporosis, which we'll talk about in just a minute. But here you see a table where we look at all of the cell types, the hormones they're produced, the stimulus that causes their secretion, inhibitors of their secretion, the target tissues, and the effects. And again, I've already talked about this, so I'm just going to let you look at it. I'd pause this video here if you need to. I'll give you a couple seconds on each slide. Again, just a second or two to screenshot this if you need it. Now, you may remember from the bone chapter, you guys talked about osteoporosis, and this is a mineral decrease in the density of bones, resulting in weak and porous bones, and it significantly increases the risk for fractures. And now one common treatment for this is a calcitonin prescription administered via nasal spray, and this nasal spray has a much higher concentration of calcitonin than would be produced by the thyroid gland. And that works to increase bone mass by slowing bone breakdown. Alternatively, there is another common treatment, and it's kind of surprising. A drug that mimics parathyroid hormone, and if you guys remember, parathyroid hormone is going to stimulate bone breakdown. And what happens here is that parathyroid hormone only actually has this effect on osteoclasts if the calcium ion concentration in the blood falls too low. And because the osteoclasts of osteoporosis patients are constantly doing that, the calcium ion levels never fall low enough for that osteoclast reaction to happen. And so what happens then is the parathyroid hormone is instead going to increase the concentration of blood calcium through its effects on the gut and kidneys with minimal effects on the osteoclasts. So it's not going to affect osteoclast activity, but it is going to increase calcium ions that are available for the osteoblasts that can then reinforce the minerals of your bones. Now, really quick, which of these is not an effect of the parathyroid hormone? Which of these is not an effect of the parathyroid hormone? It is increased osteoblast activity. Remember, parathyroid normally has nothing to do with osteoblast activity. All right, that's it for the first half of chapter 15. I'm sorry, chapter 16, the endocrine system. I will be back in the next one for you guys, hopefully by Friday.